Let's talk about cell membrane and transport today. So let's first look at the structure of the cell membrane. The cell membrane works by allowing some materials to easily pass through the membrane while preventing larger or unwanted molecules or materials from entering. This is what we call selectively permeable, as opposed to something that is impermeable, which means it does not allow anything to move through. So the cell membrane is picky with what it allows to enter or leave. It depends on the cell's internal and external condition. If you look at the structure more carefully, we did talk about phospholipids and how they create the main component of the cell membrane. We call it a phospholipid bilayer because there are two layers with their tails coming together and facing each other. So you can see the top layer has the phospholipid hydrophilic head pointing out. Let's say that this is the extracellular fluid outside of the cell. And then we have the hydrophobic tail that is pointing on the inwards. These are hydrophobic. They do not like water. They are non-polar in nature. So despite what it might look like in pictures, the cell membrane is actually not rigid at all. It has a fluid type of consistency, kind of similar to olive oil, and is quite flexible. This is why we call it a fluid mosaic model. So it is fluid in nature. We know that molecules are kind of moving, so they're kind of fluid, although they keep their form quite well. And the flexibility part of it is partially due to the presence of cholesterol molecules, which prevents the cell membrane from becoming too mushy or too solid. So you can see there in the picture below, not only is it fluid because of those cholesterol molecules embedded periodically within the phospholipid bilayer, but we also see that it's mosaic in structure because it's not just phospholipids. Just think of a mosaic in art, for example, where you take many different pieces of different colors, of different textures and materials, you put them together to create this beautiful piece of work that might communicate something. The same idea goes with the phospholipid bilayer. It is a mosaic. It's not just phospholipids, although the name phospholipid bilayer implies that. There's a mosaic of things that together work together to create the communication between cells. So we have those cholesterol molecules. We have in this picture, it also shows different types of proteins, whether it's an integral protein that runs the entire span of the phospholipid bilayer and pokes out on both sides. There's also peripheral proteins that can be partially embedded, such as the ones shown in the top, that blue globular structure, or even hanging off the end of one, as you can see at the bottom there. Now let's talk a little more about cholesterol. We know that cholesterol is a category within the lipids, and it contains hydrophilic and hydrophobic regions, as lipids would. It embeds itself within the spaces of the phospholipids and prevents small molecules from passing directly through the cell membrane. So it kind of fills up the extra little spaces between those phospholipids. In addition, cholesterol molecules also prevent fatty acid chains from uniting and forming crystals. So this is what allows for the fluidity instead of the phospholipids becoming too rigid without the cholesterol present. Let's not forget about those protein molecules. They are incredibly important, especially we'll be talking about them later in the transport across the cell membrane. But these proteins are embedded in different ways within the cell membrane. Some of these protein molecules act as special carrier molecules. So you can see a channel protein there. We'll be talking about that in a little bit, about how it helps carry molecules from the outside of the cell to the inside or vice versa. There are also protein molecules that act as receptor sites that receive hormones. And others act as gatekeepers, allowing molecules to pass through or to not pass through the membrane. Some protein molecules carry special carbohydrates, And these are called glycoproteins. So if you could find that on the image, kind of top left there, glycoprotein, you could see that there's a carbohydrate tail coming off of the embedded protein. So we call it a glycoprotein. Glyco kind of meaning sugar. And these are really helpful in cell identification. It helps cells identify not only themselves, but also hormones that need to seek the correct cell to be able to be transported into. So let's talk about transportation. How do things move through this selectively permeable membrane? So cells need to import needed materials into the cell and eliminate waste materials from the cell as well. It's important for nutrients like glucose, water, minerals, amino acids, and on, to be available for cellular processes like cellular respiration, photosynthesis, and protein synthesis. In addition, the products of these cellular processes like ATP, proteins, and sucrose must leave the cell somehow. Very small molecules and nonpolar molecules are able to pass through the cell membrane. However, larger molecules or polar molecules or charged ions will actually need some assistance in passing through the cell membrane. Okay, it is selectively permeable. So let's talk about how some things can easily pass through the cell membrane and the mechanism by which that happens, and how the ones that might not naturally be able to move across a gradient or be too large, how do those move to a place where they are required? So there's two main types of transport, and this is all based on energy requirements to be able to pass it through the cell membrane, through that phospholipid bilayer. So there is passive transport. Passive, no energy required. Okay, and when we talk about energy, hopefully there's a little ding ding that goes off in your head. ATP. There is no ATP required for passive transport. There's a picture to the right that shows diffusion as well as facilitated diffusion. Both of those cases do not require ATP. You don't see a picture, a little ATP burst anywhere, okay? Versus active transport, which is on the right of that picture, you see that little ATP molecule bursting. It's using energy to be able to actively, through the use of energy, move molecules across the gradients in some direction. So let's take a look at that. So let's first talk more about passive transport. So again, passive means does not require ATP or energy to move materials through the cell membrane. And there's three different ways that materials can move passively without energy. You've probably heard of these before: diffusion, grade 10 science; osmosis, that's specifically water molecules moving; as well as facilitated diffusion through the use of those embedded proteins. So let's first talk diffusion. Have you ever been in a room where someone far away sprayed perfume? What happened over a short period of time? Were you able to smell the scent from where you were positioned? The idea and example of diffusion, oftentimes we use this example of perfume, where if I was at the front of the class and I sprayed some perfume, slowly those molecules from the perfume would gradually dissipate from its original site of expulsion and spread slowly and evenly throughout the class, so that eventually at the end, once all the particles of that perfume have been spread apart, even the people at the back can smell it equally as those at the front. Diffusion works on the principle that molecules move from an area of high concentration, which is directly from that perfume bottle, to an area of low concentration. Once you spray it, those particles will slowly move to areas that don't have anything. That's a low concentration, okay? And when we talk about the movement, that's the concentration gradient that it's moving across from areas of high to low. This is a very natural course of movements that particles want to take. There's too much in the high area, they want to dissipate and spread out. And another example would be dyeing Easter eggs. So first you'd have a solution of water and vinegar, and then you drop different food colorings into that solution. And what you would notice is that the solution would change color as the food coloring molecules spread themselves out. In the human body, this happens across the cell. So a good example are very small molecules like oxygen and carbon dioxide. They can move through the cell membrane via diffusion. Since oxygen is constantly consumed by the cells for cellular respiration, the intercellular within the cell, the concentration within the cell of oxygen is always very low. It's constantly being consumed, so it's very low. Therefore, oxygen diffuses from the blood that's carrying oxygen into the cells. It moves without required energy, and it moves from the area of very high concentration with those red blood cells that brought the oxygen from the lungs, and it moves into an area of low concentration within the cell as it passes by. Carbon dioxide is produced during cellular respiration, and therefore, because the concentration is high within the cell, as blood drops off oxygen, it is low in carbon dioxide, and the blood will pick up the carbon dioxide and remove it from the cell. So again, it's going from the area as high concentration, within the cell to low, and it gets carried out away, and we exhale it. There's a picture down below that kind of demonstrates that concept of diffusion with blood circulating oxygen and how it drops it off in the red blood cells that are low in oxygen, and then it carries the carbon dioxide away and expels it outwards. Diffusion will end once the molecules are evenly dispersed between that internal and external cellular environment, or we call it intercellular, inter meaning within and intra meaning without, that outside, sorry. So similar idea of diffusion is osmosis, but when we're talking about the movement of particles from high to low concentration, we are talking about specifically water molecules. Okay, so with osmosis, water molecules move from an area of high concentration to an area of low concentration through a selectively permeable membrane, just like our cell membrane. In the picture on the right, you can see that there we have, I guess, a beaker, and there they put a selectively permeable membrane denoted by a red dashed line down the middle. And you can see on one side the sugar molecules, which are the green spheres, there are none. It's a very low, actually zero sugar concentration. And the water concentration is high, 100% in this case of water. Now, water is going to move from the area of high concentration to an area of low concentration. So even though there is water on the other side of that selectively permeable membrane, there is a lower concentration of water molecules in comparison to the left side. So water is going to move from where there's a lot of it to where there is less, okay? And that is the idea of osmosis. Now, relating this back to the body, the movement of water in and out of cells is very, very important to all cellular processes. Maintaining equal water concentration in the interior and exterior of the cells is very critical for cell survival. So I have examples down below of an animal cell, so red blood cells, as well as a plant cell. And let's use the terms isotonic, and iso is in both images down in the middle. Okay, this is the ideal state that cells would be in because everything is balanced. The body wants, or living things want, a balanced state. Okay, so in this case, for the isotonic of solution of red blood cells, as well as the plant cells, we see that water is equal on both sides of the cell membrane. The rate of movement of water molecules into the cell equals the rate of water molecules that are moving out of the cell. It's perfectly balanced. Now, if we look at hypotonic solution, and that's on the right side of each image, there is a higher concentration of water molecules outside of the cell as compared to inside. Inside, there's less. Okay, so where water is going to move, it's going to move into the cell. Through osmosis, it's going to go from the area of high concentration, which is extracellularly outside, to inside. And as a result, the cell starts to swell and can eventually lyse or burst. So you can see that in the hypotonic solution of the red blood cells on the right there, as well as the hypotonic plant cell. Plant cells don't burst as easily because it does have the extra structural components of the cell wall. But in this case, the cell is hypo. If you see the O, that means that there's like too much and it can burst. When the cell is placed in a hypertonic solution, so on the left, you see the red blood cells, and what's happening is that it's going to shrink. Same with the plant cell. You can see hypertonic, it's going to shrink. There is a higher concentration of water originally inside that cell. And what is going to end up happening is that the water will move out of the cell into an area of lower concentration, and it's going to leave the cell and let it start to shrivel up and shrink. And you can see that right there, which would result in a plant, in this example, low turgor pressure, and it would start to wilt. So we've spoken about diffusion, we've spoken about osmosis. Let's talk about facilitated diffusion. So this is not just transport from an area of high to low concentration of these molecules, but we're actually going to be seeing these special proteins that are embedded within the cell membrane, helping the movement of these molecules. It's still passive transport because it relies on the movement of materials from high concentration to low concentration. Okay, so that's the natural movement that particles want to move in. But we do not see the use of energy. We don't see any ATP involved in this process. The only difference is that the molecules are not able to pass directly through the membrane, perhaps based on their composition. So they require either a protein channel or protein carrier molecule to assist in the movement. Okay, so this is with the use of embedded proteins in there, in the cell membrane. A little more about facilitated diffusion. These protein channels and protein carrier molecules are very specific to the type of molecule. Each will allow it to pass through the membrane. Remember, it's selectively permeable, that membrane. So this is how it can be a little picky. These protein molecules work to bring molecules into the cell and out of the cell, all depending on the concentration gradient. A great example of this is glucose. It requires a protein carrier to enter the cell due to its slightly larger size, right? And also its polarity. So if it can't quite get through the phospholipid bilayer, it would require the use of the protein channel or carrier. Without the protein carrier, glucose would not be able to diffuse into the cell. Okay, so here's an example down below of glucose moving through with the use of sodium. Sometimes it's kind of coupled with the different molecules that needs to be moving through. So just to recap, when passive transport happens, the molecules are going to be following the concentration gradient. It's not going against it. And because of this, there's no ATP required. Okay, although it's facilitated diffusion, there is no energy required. Okay, so what in the world is active transport? We know that's simple, so diffusion without proteins, and facilitated diffusion with proteins, doesn't require energy to move a substance. But active transport does, and it uses the energy molecule that is ATP. So you can see that on the right there. Because it's moving particles against the concentration gradient, this is why it requires energy, which all embodies the idea of active transport. So active transport requires the cell to expend energy to move materials across the cell membrane against the concentration gradient. So going from an area of low concentration to an area where there's already a lot of that molecule. It's pushing it up against that concentration gradient. So materials are moving from narrowed low to high. Active transport is needed by your kidneys, for example, to regulate sugar, salt, and ion concentrations in your blood. It's also required by your muscles so that they can relax and contract, as well as your nerve cells so that impulses can be sent quickly. Active transport can bring materials into or out of the cell. It goes both ways. One way that this can be achieved is through an example here of a sodium-potassium pump. This is what specifically allows nerve and muscle cells to function. It requires energy to move sodium out and potassium in against the gradient. We will be seeing this quite a bit in our metabolism unit. And you can see that there's the A. So it doesn't actually say ATP, but the way that you can identify this one is that there's an A with 3 phosphate groups attached to it, right? That's the adenine base. 3 phosphate groups, those little white circles at the bottom of that first integral protein on the left there. Okay, so you can see that there's 3 phosphates, and in step 2, a phosphate detaches from the ATP. Now APP is, you can see the A and the 2 P's moving away. That's ADP, diphosphate. And that breaking of the bonds releases energy and allows for this protein to move particles against the concentration gradient. In this case, it's sodium and potassium. Now, bulk transport. We've been talking more about smaller molecules and particles. What happens when there's quite large quantities of materials that need to cross a membrane, or the molecules themselves are too large? So there's 2 types. There's 2 ways that the cell does this, through endocytosis, which is the way that cells bring in these larger materials into the cell through the cell membrane, as well as exocytosis, releasing large materials and moving it out of the cell. So endocytosis, cyto meaning cell, endo meaning in. Okay, so it's bringing things into the cell. When molecules are too large to enter the cell by passing directly through the cell membrane, the membrane itself formed a vesicle. It actually, as the material come in contact with the cell wraps around, the membrane wraps around the material and it brings it into the cell, and it forms a vesicle once it breaks off and enters into the cell. There are 3 ways that a cell can achieve endocytosis. So there's phagocytosis, where the cell membrane engulfs the large solid materials, as I just described. White blood cells, for example, use this process to destroy bacteria and viruses. There's pinocytosis, where the cell membrane takes in dissolved material into from the external solution and brings it in. This is sometimes called cell drinking because it's smaller amounts. And then receptor-mediated endocytosis, which involves special receptor proteins that are embedded in the cell membrane. These receptor proteins will only recognize and bond to specific molecules. Once the molecule and the receptor form a bond, the cell membrane will then fold around the complex. It's kind of like a lock, a key to a lock. And then it can enter the cell. Many hormones require receptor proteins in order to enter the cell. Here is a picture depicting all of these. Okay, so phagocytosis, there's large solid particle. They call what is pseudopodium. Pseudo meaning kind of fake, podium, poda, maybe legs. So it really wraps around it. It's almost like it gives it the big hug, that plasma or cell membrane, and it eventually engulfs it. That engulfed vesicle, the engulfed particle is surrounded by components of the cell membrane that it wrapped around. And that vacuole that is now able to move within the cell is called a phagosome, or a food vacuole. Pinocytosis, you can see it takes smaller portions, smaller particles and it says form a vesicle as well. And then cell receptor-mediated endocytosis, you can see that there are actual receptors on the surface of the cell membrane. And it takes a certain type of fit for those particles to fit into the receptor to then elicit the response to enter the cell and bring it in. Exocytosis, expelling X. So you have an ex-boyfriend or girlfriend, you want to get rid of them. Proteins, lipids, and other materials synthesized within the cell need to leave that cell. They're too large to pass through the cell membrane. They can't happen via diffusion or osmosis. They must exit through the membrane with the help of these vesicles. So it's the complete opposite of endocytosis. Now we have vesicles that do have the cell membrane around it, and it's going to be emitted into the extracellular fluid. A good example of this is waste materials that need to be removed from the intercellular space and exit the cell, and they use this process. Okay, once those vesicles are attached, that's when the materials are released